Hello, everyone. Welcome to today's live broadcast, Cell Imaging and Luminescence-Based Cytotoxicity Analysis. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and brought to you by PerkinElmer, a leading provider of multimode detection plate readers and life science lab solutions. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem to the green Q&A button lower left. I would now like to introduce today's speakers, Dr. Frauke Haenel and Dr. Philip Gribbon. Frauke Haenel studied biology and achieved her PhD in 2011 at the University of Kassel, Germany. She joined PerkinElmer in May 2011 as part of the application support team based in Hamburg, Germany. The team provides support for the multimode detection plate readers to customers, sales, and service. Within this team, Frauke mainly focuses on providing assistance on imaging and label-free applications, including training and application development. Dr. Philip Gribbon is the Chief Scientific Officer at the Fraunhofer IME, which recently integrated with the European Screening Port, EST, and which is based in Hamburg, Germany, and works mainly with academic research institutes in the area of drug discovery. The ESP uses a range of high-throughput screening, HTS, systems and assay methods and provides support for biomarker studies within clinical and preclinical research projects. Since the setup of the ESP, Dr. Gribbon has worked in close collaboration with leading life science industry partners such as PerkinElmer to help validate novel technologies which can directly apply to projects. I will now turn it over to Dr. Frauke Haenel for her presentation.
Thank you very much, Judy, for the introduction. Yes, I would like to start first with some experiments that we have done here in our lab to show what the EnSight Multimode Reader is capable to do in terms of multiparametric analysis of cytotoxicity. And I just want to quickly tell you what the EnSight Multimode Reader is, just to give you a quick overview. So the multimode reader, the EnSight, has different measurement modes. So it is included conventional label technologies like absorbance measurements, fluorescence intensity, luminescence, alpha, and TRF. We have on board the label-free technology, which is a Corning technology. And now, which is pretty new, we have the well imaging measurement mode available on that instrument. The EnSight imaging module acquires one image per well with a 4x objective. And you can see on the left, on the image, a raw image of HeLa cells. This image was generated on the EnSight system and gives you a feeling for how the images look like. And you can also see in the zoomed in part how well you can see the cells with the EnSight. The different imaging modes that are provided by this instrument are Brightfield imaging, digital phase imaging, also called DPI, and fluorescence imaging. With the EnSight, because we acquire only one image per well, we have really short reading times per plate with less than 6 minutes for a 384 well plate. So the EnSight is a very fast and sufficient method to capture all the cells in a well and actually to provide orthogonal approaches, but I will come to that in more detail later on. What we can do with the EnSight is, so this instrument allows for quickly determining the cell number and the confluency in a well, also for cell lines with diverse growth morphologies. We can detect incorrect cell feeding by looking at the cells or incorrect liquid handling. We can also detect bacterial contamination and aggregating compounds with that instrument. So it is in general a very good tool for controlling cell-based assays with the Brightfield digital phase and fluorescent imaging measurement methods. What I would like to show you today is how we can also detect cytotoxic compounds. And well, why did we choose cytotoxicity? So cytotoxicity can be caused by different types of agents, so meaning, for example, by drugs, pathogens, immune cells, and external stress factors. And cytotoxicity is still a major cause for drug withdrawal during drug development. The cell death that the cells undergo through the cytotoxic effects includes complex signaling programs that lead to a variety of different phenotypes. So, for example, you get nuclear fragmentation, cell shrinkage, membrane blebbing, or loss of membrane integrity, leakage of cell content, and swollen nuclei. So, with these phenotypes, cytotoxicity is an ideal application to be analyzed using cell imaging. And you can use that easily in addition to conventional cytotoxicity assays. And what we have done here in our lab is that we have used an orthogonal cytotoxicity approach. So we have collected different data sets from different readouts. So we have looked for the cell morphology with stainless Brightfield imaging. We have looked in more detail on the nuclei and have determined different characteristics of the nuclei with fluorescent imaging using a nuclear stain. We have determined cytoplasmic enzyme activity with the imaging mode with fluorescence imaging using an enzyme-dependent cytoplasm stain. And we have also used a conventional cytotoxicity assay, which is the level of intracellular ATP, which we have measured with the luminescence readout. And the workflow of the experiments that we have done are the following. So we have seeded HeLa cells on a view plate 384, so approximately 1,500 cells per well, and have seeded them. And on day one, we did a Brightfield imaging measurement and Brightfield analysis. Then we added DMSO, which is our model compound in this case, and incubated the cells at 37 degrees overnight. On the second day, we did also a Brightfield image analysis after the treatment. After this Brightfield imaging and analysis, we have added the stain for the nucleus and the cytoplasm, and used the fluorescent imaging capabilities of the instrument and the analysis to look for the nucleus characteristics and the cytoplasm. And at the end, we removed part of the supernatant, which was important for due to the limited volume that we had available in the well, and added the ATP kit solution, which is a kit that detects the ATP, the intracellular ATP level. And for this, we measured the cells in luminescence. Yes, and the results, so what you see here is on the left, you see a divided well. So the upper left corner is the well on day one, where you can see that you have a rather low confluency of the cells, which is different on day two, which is the upper right corner marked with A2. Here you see the untreated cells, so which means you can see how the cells just divided overnight, and the confluency is increasing. And on the right, you see a treated well. So on day one, the cells look all healthy, and on the second day, you see that the cells are shrinking based on the DMSO treatment. What we have now done with the EnSight is we have used the Brightfield confluency analysis, which is a preset analysis tool on the EnSight, and what it does is it automatically discriminates between empty image areas and the area that is covered by cells. And what you can see in the untreated well on the left, on day two after treatment, you get a confluency of about 65%. But if the cells are treated, so meaning the cells are shrinking, the confluency decreases and you get a confluency of only 29%. And if you now evaluate that and plot it against the DMSO concentration, you see on the left that the confluency decreases with increasing DMSO concentration. Remember that we have done the Brightfield imaging on day 1 and day 2. So if you subtract the confluency determined on day 1 from each well, then you get also information about cytostatic and cytotoxic effects. So meaning you can see the orange line is actually the line that distinguishes between cells that have been just stopped growing and cells that are really lysed and that are dying. So with this approach, doing imaging on one day and on the second day, so doing this kinetic approach, you can distinguish between these two effects. Another parameter that is provided by the instrument is the foreground roughness. So this is a parameter that can also be used to determine toxicity actually. So there you get, it's a texture parameter that uses the texture of the cell area, and this texture increases due to enhanced roundness of the dying cells. So this is a parameter that can also be used to determine cytotoxicity. The other approach, so you see here we have now added stains. There's two stains. So one was the Höchst stain, which stains the nuclei. It more or less stains all cells, so it is independent on if the cells are dead or not. And we've also added calcium AM, which is an enzyme-dependent cytoplasm stain. And this stain stains only living cells. And what you can see in the images is the untreated cells, they show homogeneous nuclear staining and homogeneous cytoplasm staining in green and shown in blue here. On the right panel, you see the overlay. But if you look at the DMSO-treated cells, you see that there are tiny and bright small nuclei shown, and only a few cells are positive for calcium AM. And the way we determined the cytotoxicity was that we used the preset Kaleido Cytonuc analysis. And what this analysis does is, so first of all, this analysis automatically looks for the nucleus stain, Höchst, so it detects where is a cell, where do I find the nucleus. After that, it produces, or it forms a ring structure around the nuclei to analyze the cytoplasm stain intensity of this ring. So in that case, we have analyzed in more detail the intensity of the calcium stain around the nucleus, which you can, and this is shown on the picture in the middle. And using this analysis for both treated and untreated cells, you get different analysis parameters out of this analysis. And I will now switch to the nucleus characteristics first. So what you can see here is if you look at the DMSO concentration versus the number of nuclei, the median nucleus area, and the median nucleus intensity, you see here changes depending on the DMSO concentration. And in the upper graph, you can see that the nuclei, that the number of nuclei decreases if you increase the DMSO concentration. So, and this is obviously based on cell death and the nucleus area. This is the one in the middle is based on the nucleus shrinking. So this area decreases with increasing DMSO concentration, which is just because the cells are dying and shrink. The median nucleus intensity, in contrast, increases with increasing DMSO concentrations, and this is due to nuclear condensation. This is one reason. So you get a smaller nuclei, and in addition, these smaller nuclei have a higher amount per volume, a higher amount of stain per volume, so meaning the intensity of these nuclei appears brighter. Another parameter that we get out of this Cytonuc analysis is the intensity of the calcium. So this is the parameter that is detected in the ring structure around the nucleus. And what you can see here is that the light green curve that you see is actually the labeled approach, as I have explained it so far. So we detect first the nuclei stain, and then determine in the ring structure around it, determine the cytoplasm stain intensity. And what you can see here, we get an EC50 of 0.42%. The dark green, in contrast, is a bit different. So there we have used a stainless approach. So this means we have not used the Höchst stain at all, but we have used the digital phase imaging, which produces which forms an image and which gives you the shape of the cell, more or less. And this digital phase image so detects the whole cell. And in that case, we have detected the whole cell first and determined the intensity inside the cell. And this is a nice approach because you don't need the Höchst stain and can use this detection method for cells that are only stained with calcein, for example. And you see the EC50 values with both detection methods are the same. And just to understand what the calcein AM is actually doing, so what you see with increasing DMSO concentrations, you get a decrease of calcein. And this is typical because the calcein AM is just fluorescent if it is converted by intracellular enzymes. And our orthogonal approach here was the luminescence assay that we have done using the ATP light kit. This is a kit, as I have mentioned already, which determines intracellular levels of ATP, and it's frequently used to detect cell death. What we have done here is, you can see on the left the plate map of our assay plate, and what we have done is, so we have compared wells that contain the stain of the fluorescent imaging already and cells without the stain, and added the ATP light kit on top of it. The plate type we've used is a black view plate 384, which might be surprising for the ones that are used to luminescent assays, because usually luminescence assays are done in white plates. Additionally, because we have an imaging plate with a clear bottom, we covered the plate with a white backfill to decrease luminescence crosstalk. And you can see there on the plate map you don't have any crosstalk with this backfill. And the ATP light one-step kit was added after 2 hours of the stain addition of Höchst and calcein. And what we got out of it is are the graphs you can see here. So what you see is the ATP light only, without having the stains still present in the solution, results in the same EC50 value as plus stain. So both curves are very, very similar, and what you see here is that the ATP light one-step kit is not influenced at all by the presence of Höchst and calcein stain in the supernatant. And this nicely shows that you can easily combine fluorescent imaging with the ATP light, for example. It might be, or this needs to be tested for different or different kits and different stains, but in that case, it is really nice because you don't have to take care of the stain and can look at from different angles to your cells. And to summarize all the EC50 values that we got for our multiparametric cytotoxicity assays, what you see is so most of the EC50s are pretty similar, between 2 and 3%. What is very very prominent here is the values you see in green. So here the green shows the results for the enzyme-dependent cytoplasm stain, the calcein, and here you see that the EC50 value is is reduced in contrast to the other EC50 values. And this makes sense because the calcein is known to be a very early event in apoptotic cells, so the calcium is just more sensitive to DMSO concentrations than the other readouts are. And yeah, with this approach, you see that you can distinguish between different levels of cytotoxicity using fluorescent dyes. And just to summarize, so with the EnSight, you can gain more data and findings faster out of your sample to confirm your results. You can check the cell morphology, cell nucleus and cytoplasm characteristics, as well as intracellular ATP levels. You can do that all in one well, so you get the complete picture of cytotoxicity on different levels. You can watch cell proliferation and compound effects by using stainless Brightfield confluency analysis. So this is a really robust and easy to apply approach. It's non-invasive and is lifestyle assay. And it can also distinguish between cytostatic versus cytotoxic effects. Yeah, and the power of image segmentation in the fluorescent image analysis is that you can distinguish between nucleus and cytoplasm, and it allows for more detailed analysis of different effects of cytotoxicity. And with adding a well-known intracellular ATP measurements using luminescence, you get a full the full picture, and in that case you use the orthogonal approach, which confirms your well imaging based results. Yes, so and this is it from my part, and now I would like to hand over to Phil.
Okay, thanks a lot, Frauke. Okay, so my name is Phil Gribbon from the Frauke IME here in Hamburg. Just got one or two slides just to orientate you about who we are and what we're looking for essentially in terms of screening technologies and assay technologies. I think as the Frauke is one of the largest applied research organizations in Europe. I think it has 23,000 people working there, about 5,000 work in the life science area. In our small part of it, we're predominantly interested in small molecule drug discovery. So that involves assay development, primary screening of up to 500,000 compounds in any one campaign, and profiling of those compounds in terms of confirming the efficacy in the primary and the secondary assays, but also in building up a sufficient amount of information to let you understand what the off-target liabilities of some of the compounds might be, in order to be able to then remove compounds from the workflow which maybe have cytotoxic effects and which you don't want to advance into further optimization. The majority of the people are coming from either industry or biotech backgrounds. So in terms of the assay systems, and again, this maps onto what we're looking for from reader systems. We have a lot of academic targets, and that means that you're often faced with some quite difficult and challenging mechanisms to investigate and to find modulators for, particularly in the areas of protein-protein interactions. So we tend to do a lot of HGRF experiments, a lot of alpha-screen type experiments, along with the types of assays where you would have luminogenic or fluorogenic substrates, for instance, kinases and proteases, etc. We're also interested in phenotypic as well as target-based readouts. And what I'm going to go through today is really an application of the EnSight in a phenotypic assay format, where we're looking to characterize compounds coming from a marine fungi source and trying to understand what their anti-cancer properties might be. So it's not really from the pure toxicological perspective where you're eliminating compounds, but really we're looking for compounds which have specific patterns of toxicity against cell lines which are well characterized and well defined, and which we source from the NCI panel in the National Cancer Institute in the US. So just to, our workflow is very similar to that was characterized by Frauke. And what we wanted to do is we had over 1,000 compounds, and we were looking to profile those compounds against up to 35 cell lines from the NCI panel in a phenotypic assay, where we're looking for compounds which should inhibit the growth of the cell lines, and compounds which would also have cytotoxic properties. So we do cell seeding and then add the compounds or control compounds of DMSO after 24 hours, wait for 48 hours, and then our classical workflow before we really had access to the EnSight and other imaging techniques was to do a viability detection using cell titer glow or ATP light or other types of metabolic readouts using ATP as a surrogate for cell number. And that allows you to calculate from the dose response data the cytotoxic effect and the cytostatic effect as well. So this is again along the lines of what we've seen with Frauke's ATP light, and here we're using the cell titer glow from Bromiga. I think one thing that's worth pointing out for all of these metabolic cellular metabolism readouts is they can be extremely sensitive. So you can get down to very low cell numbers in certainly the hundreds, but in some cases the 10 to 100 cells is pretty accessible using some of these quite sensitive detection technologies. So if you're looking for complementary technologies, then you need to be thinking about things which have similar levels of individual cell-based assessment. So they are quite good technologies. I think if you do have to do a reasonable amount of work, and we had 35 cell lines that we wanted to use in our panel, and our intention was to screen 1,000 compounds in triplicate and 11-point dose response. So there was quite a lot of work to do in terms of assay development. So we're doing the classic assay development here again for the cell titer glow. So if we're looking here, we're doing the DMSO tolerance, and you can see DMSO was used in a cytotoxicity standard. You can see we want to keep well below 1%. And we're checking the linearity of the detection in terms of the number of cells. And we had a panel of standard compounds where we knew what the expected pharmacology would be, and they were cisplatin, mercaptopurine, storosbon, and paclitaxel. And also we were running some experiments whereby we wanted to see whether there was some counter-assay effect whereby the compounds were affecting the detection reagents. And we also picked up some hints of that for some of the compounds. Again, it's a slight diversion, but what we're seeking to do with these phenotypic assays is, obviously there's an underlying target that we're hoping that the compounds will be modulating in some beneficial way. The problem we have is that we don't know what that target is in many cases. So the NCI offer quite a nice tool called the compare analysis, whereby you're able to look at the profile of activities of your compound and upload that into a database where it's then compared against the profile of activities against different cell lines of standard compounds, so standard kinase inhibitors, HDAC inhibitors, etc. It can use a correlation analysis, and I think there's 38,000 compound profiles available in there. And that will give you a first indication of what the mechanism of action may be of your compound, if it matches or at least correlates in some way with the profile seen for the standard compounds. And you can see here that it can be, this is a result for cisplatin, which is one of the standard compounds that we used across all of the screen here. And cisplatin is a DNA cross-linking agent. And you can see here when we compared cisplatin against 171 compounds which are found in the standard drug set on the compared database, that we get very good correlation. And in terms of the rank, cisplatin and cisplatin-like compounds are highly represented in the top series. If we look at the 38,000 compounds in the entire database, you can see again that cisplatin and compounds which have similar mechanisms of action and DNA cross-linking to cisplatin, again, are highly ranked. So I think this is just a message that I think the existing technologies are very good. There's good databases out there that can help you understand what the preliminary mechanisms of action and what your compounds might be. So everything looks rosy in the garden, but it isn't really the case. I think we have to look at where there are some problems with ATP detection. It's certainly not a universal readout, and there's a growing body of evidence that certain cell growth and toxicity mechanisms are not adequately represented in this metabolic readout. And essentially, these are compounds which can either increase the size of cells or increase the mitochondrial content in cells, and they could give you, they could misinform you with respect to the potential cytotoxic effects of your compounds. I think another thing which is missing, and there are some new technologies which are coming on board in the steady growth systems, but certainly in the classic cell type growing ATP light, is the fact that you need to allow your cells to release the ATP in order to be able to run your measurement. And this means you've got a one-time endpoint measurement. And I think that whilst that's very good and can be sort of helpful in terms of throughput, actually you're missing a trick, so you can miss a trick sometimes. Some of the mechanisms of action of cytotoxicity can appear on the timescales of minutes or hours, particularly, for instance, membrane disrupting reagents, detergents, etc. Whereas the cytotoxic effects of other compounds can happen over many days. And it can be of use when you're, again, triaging your compounds to try and understand what the kinetic component of the cytotoxicity is for particular compounds, and then using that as a filter or not in selecting for compounds. And also, you know, these reagents are relatively expensive, so it is an additional detection reagent, so there may be an opportunity to reduce costs. So this is where we were coming from as an organization, using these types of technologies on a day-to-day basis. So I think we've had an explanation of the way the Brightfield illumination works from Frauke, and having the EnSight on board has allowed us to see to what degree we can transfer our pre-existing ATP-based readouts, see if we can transfer those to a non-label, let's say, rather than label-free, but a non-label, non-stained, non-additional detection reagent required readout for cell viability. And so the key parameters, I think, have already been mentioned. These, I think, what are, apart from the general confluency parameters, which are sort of telling you how much of the surface is covered by cells, there's also a number of very interesting parameters which really indicate what's happening on a very localized area within the cells. And these are based upon the roughness, and they're based upon local changes in intensity. And we've been really just experimenting, playing around to a certain degree to try and understand, using the pharmacology of known compound standards to guide us, to try and understand what these parameters are telling us about the health of the cells at any point in time. So we've got these various parameters, and I'm going to look at some of those now and see which may be useful and which are not quite so useful, and which ones may be even giving us early indications, in the same way that the calcium dye mentioned earlier on, early indications of cytotoxic effects within the cell. So I think the basic parameter that you need to be able to determine in these imaging-based methods is confluency. So here you can see for two cell lines, for the MCF7 and the 7860, that we can measure changes in confluency quite successfully over different time periods, without seemingly adversely affecting the cells. The measurement time per plate is relatively low, at less than 4 minutes at this basic readout without any fluorescence channels. And we can use that as essentially a quality control measure within the screen to ensure that the number of cells that we have is consistent across the number of wells within a plate, and that our dispensing systems are working adequately. When we measure confluency in the presence of DMSO at 0 and 5%, and also paclitaxel, you can see there that you're beginning then to see changes. So where you, at the top one here, where you just have 0.5% DMSO, the confluency increases as expected. At a higher percentage of DMSO over time, up to 72 hours, you're seeing that the cell growth is being stunted here. So it's a cell growth inhibition. And you can see for paclitaxel at 5 micromolar that you're seeing a cytotoxic effect with time. So these are all basic parameters. if we're now moving away from the coverage of the cells, actually looking at the local roughness parameters and the local texture-based parameters associated with the cells, here's another couple of examples here. Now you can see here at high DMSO concentration that you're getting an increase in the roughness. And you can see again, similar with the, this is high paclitaxel cell concentration, it's causing an increase in roughness. But at your 5% DMSO, you're seeing no change there, and at your low DMSO concentration, you're seeing a reduction in roughness. And this is indicative of the cells, healthy cells spreading along the bottom of the plate. And the same sort of parameters I've seen with MCF7, so you're getting an increase in this fraction of high contrast cells. You're seeing no changes with 5% DMSO, and you're seeing a reduction in this fraction with at low DMSO concentration. So the roughness-based parameters and the contrast-based parameters are giving you additional information independent of the confluency-based parameters. And it seems to be the changes, the relative changes are larger than we see with the confluency-based parameters, which gives you some help, like some indication that you might have an orthogonal imaging-based readout describing what's happening with the cells. Here's another parameter. In this case, it's the foreground roughness. You see here an increase in paclitaxel. It's not changing in the presence of 0.5% DMSO. So we can pan our way through all of these different readouts. In this case, we've got all of the different readouts here, and we're looking at increasing concentrations of colchicine. And you can see here with this particular parameter, which I think I can't read on my screen, is the my eyes are getting old. Yeah. So this the confluency here. This is the high contrast region roughness. This is the roughness range factor. This is the foreground roughness, and this is the low contrast roughness range in the mean. So you can see that there's significant changes in these texture-based parameters associated with increasing concentrations of colchicine. And in some cases, such in the roughness range factor, you can see that the relatively healthy cells, which are exposed to very low concentrations of colchicine, they're seeing a reduction in these parameters, whereas the cells which are exposed to higher concentrations of colchicine, you see an increase in these parameters. So it's quite a nice assay window which is opening up in those cases, which is in comparison to the confluency measurement, where you're just seeing either an increase relative to the baseline. Again, you can see here we've taken 6 standard compounds. 2 of those compounds are non-cytotoxic, which carbamylcholine and pilocarpine, and we've got colchicine, paclitaxel, trichostatin A, and gentian violet. You can see quite nicely for the paclitaxel and for the trichostatin A that we're seeing quite nice dose responses forming, but not fantastic, I would say, with the confluency. But then if you compare the same sets of compounds using the foreground roughness mean, you can see quite nicely developed dose responses, particularly for I think the paclitaxel here, you see very large windows becoming apparent, and also for the trichostatin A, there's nice windows being formed here. We did see some problems with the gentian violet, and I think that in a sense that might be because of some optical interference between this, which is it's a colored compound, and that's something that we're looking at into more detail on whether we can remove the effects of colored compounds and their influence on these parameters. We can see the same thing with the roughness range factor. Here, the dose responses are less pronounced, and this in our hand seems to be less of a useful derived parameter, but you can still see substantial dose responses. And then we can compare finally with the the cell titer growth from Promega. You can see here you get nice dose responses, but again they're not too dissimilar to what we observed with the range factors, the roughness factors. So in terms of, if we can summarize the data for ATP growth versus image-based analysis, these are all the IC50 values expressed in micromolar. You can see here for our standard compounds at 48 hours, which was the last set of data, that we're getting reasonable values with the luminescence readout. But then again, if you compare that to the confluency, you can see again a reasonable correlation here for colchicine, and also for the roughness, the foreground roughness parameters, and the roughness range parameters. So we seem to have very nice correlation in some cases for some compounds between the luminescence, the confluency, and these roughness-based parameters. There are some cases where no fit was possible, particularly for the confluency and some compounds like trichostatin A, for instance. But where you weren't able to get a fit with confluency, you were actually able to get quite a reasonable fit using some of the roughness parameters. So you see a good correlation between the 0.037 and the values that you get in foreground roughness and in the roughness range as well. These correlations are also borne out in a second cell line, in the 7860 cell line. So we're by no means explored all of the possibilities when it comes to understanding what the different effects of these compounds are on these roughness-based parameters as measured using the Brightfield illumination. But we certainly see a strong indication that there's a good correlation between what we see with luminescence and what we see with optical-based measures of confluency, and then also what we can see with these roughness-based parameters, the foreground roughness and the roughness range. So in terms of the overall performance, we think that certainly in our hands so far, Brightfield imaging seems to produce results for confluency which are consistent with what we've seen with ATP determination. These morphological or roughness based features seem to give us at least useful secondary orthogonal readouts. We're able to read the plate relatively rapidly, less than 4 minutes per plate, and what we don't have it in our shop is we don't have access to environmental control of CO2, etc., within the confines of the reader. So we're essentially taking the plate out and reading it straight away. And I think these rapid read times and comparatively low numerical aperture of the objectives allow us to quickly read the plates and then put them back into their environmentally controlled incubators, and then we're not perturbing them excessively. We've actually taken around 10 cell lines to date, and we've been able to measure these optical-based texture parameters for those cell lines reasonably successfully. It does require a certain amount of tuning when it comes to defining the parameters within the algorithm for identifying the cells and defining the properties of the cells. But we haven't had a cell line so far that we weren't able to usefully extract these parameters from. So where are we going next with the EnSight? We want to extend the image analysis process to cover additional cancer cell lines. We want to see whether we can replicate some of the data I showed earlier on when it comes to the compare analysis to see whether we can use these optical-based parameters also as an insight into potentially what the targets may be when we're screening multiple cell lines in parallel. So that's something that we're looking to do and getting the cell lines in place to do that. We want to look at some of the more problematic compounds which have been shown to, in recent papers, to cause enlargement of the cell and increase in the proportion of mitochondria in each cell, and see whether we can get more reasonable cytotoxic readouts for those compounds. In the medium term, our goal is really to see whether we can replace ATP determination with imaging-based assays, and we want to be able to measure effects on growth and effects on cytotoxicity, and that, in a sense, requires that you're able to differentiate between a live cell and a dead cell. And I think we're not quite there yet. I think we'll get to that point quite soon, and that's something that we're working very closely with the team in PerkinElmer to achieve. So just to give you an overall impression, we've had this system in our lab for over 6 months now. We see the advantages in terms of having access to an orthogonal technology, and that's always critical when it comes to validating the hits which are coming out of screens, because so many compounds can cause artifacts and cause you problems. What I haven't really talked about today is the potential to normalize the data. That would require, I think, if you're screening many cell plates today, it would require an integration whereby you placed your EnSight into an automated workflow, where you could use that then to ensure that the number of cells that you'd seeded per well met your quality control standards. And I think that's an important additional quality control feature of these imaging-based systems. And they could be used like that in routine screening as a pre-screen. Are the number of cells in there correct and to the standard that we expect? I think that there's a lot of potential associated with using kinetic assays and interrogating the cytotoxic effects of compounds at very short and very long time scales. And the ability to do that without having to lyse the cells, I think, is very good. And that could give you the potential to rank compounds according to their kinetic response. So you may be looking for compounds which have a rapid onset effect, or you could consider that many of those compounds may in fact be membrane disrupting compounds or detergent-like compounds, and they're ones that you want to take out of your workflow and remove them from your hit list. We haven't fully got to the point where we can lower the cost for detection reagents. I think that's an aspiration in the medium term, once we're completely secure in our understanding of how the system works when it comes to what these parameters are telling us. But it could be that in the future that we can do label-free or no-label imaging of cells, and that could be the one and only readout. And I think from a general perspective, the instrument is pretty straightforward to use, and we are also users of the EnVision and the EnSight here, and it works along similar principles. And it's quite possible with the support of the team here to be able to adjust your image analysis parameters and tune your system to cater for the needs of additional cell lines as required. So I think that's everything for my presentation. I can hand back to you, Judy, for your next moderation.
Thank you. Thank you for the informative presentation. During the question and answer session that follows, 2 poll questions will appear on your screen. You may select an answer or close the poll question by clicking on the X in the right corner. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, if I obtain so many different parameters and readouts from my experiment, how do I know which of these is the right one?
Yeah, I mean, I think in that sort of case, you need to be guided by the pharmacology and the performance of standard compounds. So if your standard compound is the particular parameter, like the foreground roughness mean, for instance, if that is giving you pharmacology which is consistent with what you're expecting from that compound, and similar to what's reported in literature, then I think that's the one that you should be investing your time and effort in as your final readout. So it always, I think, as in any screening thing, it always comes back. If you've got good standard compounds to help guide your assay development and guide your quality control through a screen, then that's always the best thing to have. Sometimes people don't have that, but on the whole, that's what people's ambition is at the start of the screening campaign.
Your next question is a 2-parter. How does the image analysis work, and do I need any One second there. Do I need any special training for the analysis?
No, you don't need any special training. So the EnSight is equipped with Kaleido, which is the analysis software, and there you have different methods that I have shown also in my presentation, and you just choose one. You have a few parameters that you can adjust based on your application and cell type, and then you just go. So there is no training needed, so it's really straightforward.
What colors can the EnSight use for fluorescent imaging?
The EnSight is equipped with up to 4 LEDs, and we have LEDs for, from the excitation side, we have LEDs for UV, blue, green, and red. And on the emission side, we have a multi-band filter, which allows for simultaneous emission detection. And this is actually the reason why it is so fast. Because on the emission side, we don't have to switch any filters.
It was mentioned that the EnSight takes less than 6 minutes per plate. Is this measurement time dependent on the number of excitation sources?
Yes, it is dependent. It's always dependent on the settings, because you might use dyes that need longer excitation times because they are rather dim. Here I would like to mention, so if you have more than one channel added to your protocol, it does not mean that the measurement time doubles. It is approximately 20% that the measurement time increases. And this is based because you have different movements, for example, the movement of the plate, and also the time the autofocus needs, that are just done once per plate, and then you have just the additional time for the second channel, but it's not doubling.
Is there evidence that texture-based analyses are more sensitive to early cytotoxic effects than confluence or metabolic readout?
I think there's some preliminary evidence that for some compounds it seems to be more sensitive. I think we have to do a more comprehensive assessment of that, and so that's a work in progress, as it were. But certainly they're showing promise so far as being able to, as indicating early stage intracellular events before confluency or before metabolic readouts.
Did you use coated view plates?
No, we didn't. For the HeLa cells, we didn't. Did you use one?
No, we don't. Yeah. We don't use plate coating.
But it's not a, yeah. You can use them as well. So.
Was ATP light reagent added without the washing step before?
Yes, and that's what is interesting, because we just removed a bit of the supernatant, which was really just because otherwise we did not have enough volume in the well, but then we added the ATP light without any washing. It was just added like that.
Yeah, and I think with the cell titer glow reagent, again, we used exactly the same plates that we had been imaging with the EnSight, and we just ran the regular cell titer glow protocol, adding, I think it's an equal volume of the cell titer glow reagent, and then reading.
The sophisticated imaging technology seems to be geared toward adherent cell lines and would replace classical assays, example MTT or XTT. How is applicability for cell-mediated cytotoxicity assays, especially in suspension cultures where effector and target cells that are often in clusters, to be analyzed?
I think that's a really tough one. In our experience, we tend to stick to adherent cell lines in many of these cases. But you do see with non-adherent suspension cell lines that eventually the cells do fall to the bottom of the plate. It's just that they don't adhere, and they tend to cluster and clump as well in a little circle at the bottom. So I think at the moment, you may need other technologies. I think if you're working with suspension cell lines, and I think there's a range of other technologies there, but it's that is an issue. I agree.
Or you would probably use stains for the suspension cells, because I mean it's obvious that the Brightfield is not very effective because suspension cells are around anyway. And but you could use like toxicity stains and use that for suspension cells.
Yeah, I mean, I have to say we've used it. We haven't presented the data here, but we've used it in some IPS lines and some primary rat and murine lines that we've used here, and it's been very successful in those as well. So it's not just these over-engineered HEC, HeLa, MCF7 type lines that it's used so far. We've used it across a whole range of IPS, human IPS drive lines, and it's been quite nice, especially in combination with the fluorescence. And I think that will be in a future presentation when we talk through that data. But that's quite nice.
Which liquid acoustic instrument is used?
For adding the compounds, in our case, we use an echo. And I think for a variety of reasons, because you want to keep for the cytotoxicity, you want to keep the compounds in suspension in DMSO, in solution in DMSO for as long as possible, and before adding to the plate without cells. I think all of the other liquid handling is it's standard Janus techniques, or we seed using multi-drops, etc. So there's nothing special about the liquid handling. It's the types of instrumentation that you find within a regular drug discovery lab, or a lab which has got a TCAN or similar types of robotic systems in place.
Can one differentiate between different cells in a heterogeneous culture system?
Yes. Yes and no. So with the current scripts, we have seen some exciting data for the Brightfield confluency analysis, actually, because you have there in the settings you can adjust the contrast threshold, and with that you can to some extent distinguish between two different morphologies of cells. Additionally, we can provide custom scripts for the EnSight, so this would also be a possibility to adjust the method that we provide on the instrument to make that possible.
Compounds that induce cell cycle arrest might lead first to swallowing of cells before they actually undergo apoptosis. Would that be a right guess to compare ATP-like versus any phenotypic assay?
I think in those types of compounds, it would be extremely interesting to get a kinetic assessment of what's going on. So looking at 1 hour, 5 hours, 10 hours. And I think that can give you a lot of information about what's happening. Every compound potentially has a different effect. But I think that's where the imaging-based methods have a real advantage over simple endpoint methods.
Does the media color interfere with detection analysis? So
I think you can't use pH dyes with the cell titer glow. I'm not sure what the ATP like, but I don't think it interferes with the imaging itself. But it does interfere with some of the luminescence reagents. So that's something to be careful of. But with the imaging itself, it's not really in our experience.
I think we may have run out of time for answering questions. Give me just a second here and just confirm this. Let's see. I think we have run out of time for questions. So I would like to thank our sponsor, PerkinElmer, for making today's educational webcast possible. There were a number of questions we did not get to today, so questions we do not have time to answer will be answered via email. Today's webcast will be available for on-demand viewing through November 26, 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.